Hi everyone. Okay, so today we are going to discuss about the structure of chromosomes. So let me just check if I'm on. Just a second. Okay. All right. Okay, so let me introduce myself. I'm Vani Kari, biology educator at an academy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Okay, so join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology. Okay, and the Telegram channel Unacademy ICSE and ISC both. So this link you will find in the description. So you can click on the link and join the channel for updates regarding the classes. Okay, so we also conduct special classes on Unacademy platform, so that is absolutely free. So learn from your favorite educator for free, engage with educators via polls, clear your doubts in live special classes. Okay, so for the top special classes you can visit the link in the description. Okay, so Unacademy subscription features, you can learn live from the comfort of your home. Okay, unlimited access to all the courses will be there. Top educators of India are going to take the classes in one platform, that is Unacademy platform. Regular doubt clearing session, so after about every 3 session, the fourth session will be your doubt clearing session. Okay, so any doubts you have related to the topics that are previously covered, so you can clear your doubts in this particular session, that is the fourth session. Okay, then complete syllabus will be covered, mentorship and guidance is given, study material in the form of PDF is given, practice tests is given, live test series, batch courses. So you will have access to all of this once you take the subscription. So there is daily practice section, also weekly mock test series as well, live quizzes, daily MCQ and subjective test series also you will have access to. Okay, so now Unacademy plus subscription features. So since you are in class 10 now, so you can try, like, how exactly this works. You can try with the 3 months also. So if you are, those who are in the beginning, like, just in the beginning of class 10, so you can try with 12 months of subscription. Okay, so for 12 months it is 15k, and by the application of code, so with the applying of code, okay, so that is Vani YT, so it will get reduced to somewhere around 13.5k. Okay, so you can make use of this particular benefit, apply the code Vani YT, okay, and get the subscription so that you can complete and class 10 with Unacademy. So all your subjects will be covered, so mainly the maths and science. Okay, so now moving on to the introduction part. Okay, so what exactly are we going to study today? It is about the structure. Okay, so structure of chromosome, how exactly this DNA is arranged in a cell. Okay, so now DNA is the basic genetic material for most of the living organism, isn't it? So DNA is the genetic material, right? Is it, or is there anything else other than DNA for as a genetic material? So DNA is the one, that is deoxyribonucleic acid. Okay, so DNA is the genetic material. So how does the length of the DNA fit into the space of a nucleus? So that is around 2.2 meters of a DNA is there in each cell. Okay, so this is in each cell, that is mainly it has to fit not in cell, but it has to fit in a nucleus. So you can imagine that such a big DNA, so how exactly it is fitting into this cell nucleus. So it's not just the cell, so it is the nucleus. So you can just imagine this is a cell now, only nucleus is this much, and 2.2 meter long DNA has to fit in here, that is in the nucleus. So how is that done? Okay, so we will study the structure of chromosome, so you will understand how exactly this DNA is fitting into this tiny nucleus. Okay, so how does it replicate to transfer the genetic information? So we will see that as well. All right, so now nucleus, so where exactly do you find a nucleus? So nucleus you find inside a cell. Okay, so now this is a cell. Okay, so in this cell you find a nucleus. So inside this nucleus you can see here, so this one is actually the nucleolus, so you can find a nucleolus as well. Okay, so now this space is actually having a cytoplasm. Okay, so there is a cytoplasm, and this one is nucleoplasm. So suspended in the nucleoplasm you can find a chromatin fiber. Okay, so there is a chromatin fiber. So this chromatin fiber is going to condense. All right, so this will condense. So as the condensation occurs, so this will condense to form chromosome. Okay, so earlier it is a chromatin fiber. So whenever cell division has to occur, so this cell has to undergo division, so in that case this is going to condense the nucleus or the chromatin fiber. What is there? So that is going to condense to form a chromosome. Okay, so chromosome structure we can see in here. Okay, let's see this one. All right, so let me just explain this part. One second. Okay, so yes, okay, so now you know chromosome, what exactly is this one? So now let's see this chromatin fiber. Okay, so how exactly you know this DNA when we mention, so chromatin fiber, which is this, so that is coiled and supercoiled. Okay, so this I'm talking about the chromatin fiber. Okay, so you know there you can find associated structures. So you can see coiling of this structure. So now this we call as a coiling. Okay, so this is actually what, so this is called as a coiling. All right, so this chromatin fiber associated with certain protein. So we will see what is nucleosome model also. So associated with protein you can see the coiling there. Then you can also see that there is a supercoiling that can be seen. Okay, so now this is a supercoiling that we see. So this was earlier, so this was just coiling like this. Okay, so then you know when the DNA is getting coiled, so this is supercoiling. Okay, so supercoiling also you see. So now this part which is here, so this is supercoiling. Okay, so now how exactly is DNA is arranged? So DNA is arranged is that is about 2.2 meter DNA in this nucleus. So that is arranged by coiling and supercoiling, so that whatever amount of this DNA is this, that is going to reduce as coiling and supercoiling is seen. Okay, so now with respect to that, one second, just a second. Yeah, so now with respect to that, so that is how the DNA is gonna be arranged. So now you know the nucleus is bound by two membranes. So there is a nucleus. Okay, so nucleus when we mention, so there is a outer membrane. So you can find there is outer membrane as such. All right, so let me just draw it here and explain. So now this one is the cell. Okay, so this cell is having what? So there is a outer membrane. So this is now a outer membrane, and then you can find a inner membrane. So there is a inner membrane as well. All right, so now outer membrane and inner membrane together, you can see these spaces here, right? So this is a pore. All right, so this is a nuclear pore. So nuclear pore when we mention, so you know nucleoplasm, so here you can find nucleoplasm, and here you can find a cytoplasm. So nucleoplasm is in continuity with the cytoplasm, so that is through the nucleus. nuclear pore. Alright, so let's see this one. So nucleus is bound by two membranes, so that we call as a nuclear envelope. So that means outer membrane is there, okay, and then you can find the inner membrane there. So there is outer and inner membrane, so together we call them as a nuclear envelope. Now nucleoplasm, so DNA, the liquid region inside this nucleus contains the nucleolus and then the chromatin. Nucleolus is responsible for ribosome synthesis, okay, so this is responsible for ribosome synthesis. Now chromatin, when we mention, so this is thread like mass, so you can observe a thread like mass which is mainly having a DNA. So that means the coiling structure which you observed, so that is the DNA along with the protein. Alright, so we will see what exactly is nucleosome. Any idea what is nucleosome? Any of you? So what exactly is nucleosome? Alright, so now you know nucleosome when we mention, so this is thread like mass. Alright, so one second. So this is having a loose connection. Alright, I think I can write now. Yeah. So yes, so a nucleosome model is basically a DNA where the DNA is wrapped around a histone protein. Okay. Alright. What is this, Piyush? Can you please speak a bit louder, ma'am? So am I not audible? Is there a voice issue? Yes, I'll do that. So I think you can, so I'll just refresh this. Okay, anyway, yes. So now nucleolus. So this may be, you know, we can find one nucleolus or many. And, you know, main function of nucleolus is to synthesize a ribosome. Okay, so let's see this one. So now this particular cell which is there. Okay, so you are not using headphone. Okay, so yes, sure, I'll speak louder. Okay, so now this one is a cell. Okay, so you can observe in this cell, so there is this part is nucleolus. Okay, now this part you see here, so this is actually a chromatin fiber which is there. Okay, so now this cell is actually getting ready to undergo cell division. So when the cell division has to occur, so you can see that the centrosome which was there, so that is divided into two. Okay, and then what happens, this starts to move towards the pole, and you can see that this particular chromatin fiber which was there earlier over here, so as the progress occurs with respect to centrosome moving towards the pole, you can see here this chromatin fiber has condensed to form a chromosome. So you can see this chromosome structure here, right? So this chromosome structure is basically condensed chromatin fiber. Okay, so chromatin fiber is going to condense. Okay, so when this condense, so it is going to arrange itself as a chromosome. So what exactly will be the chromosome having? So I'll just say this. So chromosome, when we mention, so you know it will have a outer covering as such. Okay, so there is this protein outer covering is there, so this is the primary constriction here. Alright, so now this DNA which is there, so this is coiled, supercoiled. Again you can see coiling, supercoiling. So this is the chromatin fiber itself which is condensed. Okay, so this is completely condensed. You can see coiling and supercoiling of a DNA. So that means here inside, so this is now a proteinaceous covering, or we can just write it as a protein covering is that. Okay, and inside that you can find a nucleosome. So nucleosomes are arranged, that is by coiling and supercoiling. So what exactly is nucleosome? Anybody has any idea? So what exactly is supercoiling, or nucleosome? So you know you can find a coiling and supercoiling. So this side also it is the same thing. Now this part, what is there? So this we call as a one side we are representing here. So now both, like on the whole, this one is a chromosome. So on the whole, this is a chromosome. So only part of it that we are considering. So this is a chromatin. So this side also, it is actually the DNA itself, that is chromatin fiber itself, which is again coiled. Then you can see supercoiling, again coiling, supercoiling. So this is how you can see arrangement of this DNA. So now if I try to pull out, so you can see this one, okay, I had written it is having a DNA. Yes, very good. So it is having a DNA, nucleosome. So along with that, so nucleosome when we mention, so it is not just the DNA that is present. So it is having a protein. Okay. So protein is basically a histone. So histone protein is associated with the DNA. Okay, so DNA is having a negative charge, histone protein is having a positive charge. So there is a perfect, or we can say proper binding that occurs. So it looks like this. Okay, I'll just show you this. So it is now, this is a histone protein. Okay, so this is a histone protein. So now when DNA is coiled around this structure, so this looks as though there is a bead on a string model. So let's say here one more histone protein is there. So this particular DNA is coiled around this. So there is a structure. So now this bead is there and DNA is there. So this bead is actually the histone. Okay, and this one is the DNA which is coiled around, and it is linking. So now this is also called as bead on a string model. Okay, so or this can also be called as a nucleosome model. Okay, so in general we can also call them as new bodies. So this is a nucleosome model. So I'll just explain you how, where exactly this is present over here. Okay, yes, positive and negative charge yahan bhi. So biology says here. So biology is basically, chemistry when we mention. So everything, each and everything is having a carbon, hydrogen, then nitrogen is there. So it is not something other than chemistry. So now when we study biology, so all the compounds in biology, what we mentioned, so all those are having these compounds, carbon compound, hydrogen compound, so then there is oxygen, nitrogen, so everything is there. So it is basically chemistry is related. So whatever you are studying, so that is with respect to bio mainly. So you are basic structure is these things only. Okay, along with that the phosphorus also will be there. So let's see this one. So bead on a string model also we call, okay, or nucleosome model we can say, or one more in general they also call these as a new bodies. Okay, so new body is for nucleosome body. All right, so let's see this. So I'll just explain this part. Okay, so now this is a chromosome. Okay, so what exactly is chromosome having? So this is having coiled, okay, and supercoiled chromatin fiber. Okay, so coiled and supercoiled chromatin fiber is there. So you can see here, I'll try to pull. So let's say Piyush is trying to pull this a particular thread. So you can see a particular thread is being pulled from this chromosome. So that thread is nothing but, so when you try to separate that, so this is actually the chromatin fiber. Okay, so this one is, so this is actually chromatin fiber itself. All right, so chromosome is made up of chromatin fibers will be there. So now when we separate that, you can see that there is supercoiling, you can see coiling, you can see what is bead. Yes, I'll explain that. Okay, then you can see a nucleosome there. Okay, so now you see the structure here, right? So you can observe it in your book also. So there is tiny bead, there is tiny balls you will see. Okay, so this is actually the tiny ball like structures what you observe here. So that is mainly the nucleosome model. So I'll just draw it here and explain. Okay, so now this bead what I'm talking about, so that is a histone protein. Okay, so histone protein is basically not just one type of protein you will find. So mainly you can find 5 types there. So one is H1 is there, so H2A, H2B, then H3 and H4. So these are the 5 different types of histone protein that you find in case of a DNA. Okay, so this is with respect to histone protein. So histone protein is, it is forming a octamer. So there is a formation of histone octamer that you can see. Okay, so now this histone octamer is one that is arranged as a octamer, and coiling around that you can find that there is a DNA. Okay, so histone proteins are responsible. Yes, so that's right, because histone is positively charged because of some amino acids that are present. So this is a protein. So protein means what? It is made up of amino acids. Okay, and these amino acids are actually, you will find two types of amino acids. So there is adenine, lysine is them. Okay, so that is providing a positive charge to this protein. Okay, so now this histone octamer, so you can see this is one histone protein, this is one histone protein. So here is another histone protein, and then there is another histone protein. Okay, so now this is going to form a octamer. So this is only front side I am drawing. Okay, so back side there is one more set as such. So now this is H2A. Okay, so on the back, so there is H2A again, so two units of each. So H2B is here. So on the back side again you can find a H2B. All right, so same way there is H3 here. So this one is H3. On the back side you can see H3. So now this is H4. So towards the back, so you can find a H4. So now this whole thing which you see here, so I'll just put an outline for this. So now this whole of a thing what you see now, okay, so this whole of a thing is actually a histone protein that we call in general. But this is not just one protein. So this is exactly, this is made of many different types of protein. Mainly 4 types of proteins will be there. So that is why we call this as a histone octamer. Octamer means what? So 8 of them are there. Okay, 8 of the histone proteins. Octa is 8. Okay, octopus, right? So octopus is also 8 feet will be there. So same way histone octamer is 1, 2, 3, 4, 5, 6, 7, 8. Okay, so 8 of them together. So this is going to form a bead, or this is basically histone octamer. Then there is a string. Okay, so that string is nothing but the DNA. So DNA is going to coil around this structure. So there you can see a coiling. So DNA is coiling around this one, and then there is continuity. Okay, so now histone proteins are okay. So this is the previous one. Yes, so now in this case, now this is the DNA. Okay, so this DNA is coiled around, so or we can say wound around this histone, that is a bead. And now this is a DNA, so this can be considered as a string also, and hence the name nucleosome model we can mention, or this can also be called as a bead on a string model. Okay, so bead is there and a string is there surrounding that, so we can also call it as bead on a string model, or this can in general can also be called as a new bodies. Okay, that is nucleosome bodies. Is this clear? Yes, any doubt, Piyush? So you can see there is nucleosome. Okay, so this nucleosome, so now as you try to separate them, so this was a supercoiling that you could see. Okay, then you can see this was a coiling which you can see here clearly. I'll just erase this part, just a second. Okay, so now you see this coiling here, right? So there is a coiling that you can see at this point. So now this is now a supercoiling. So coil structure is only supercoiling. No, it is not clear. You want me to repeat this? Okay, so now there is DNA. All right, so DNA is there and histone octamer you can find. So surrounding a histone octamer, DNA is there. All right, so now if we try to separate that DNA and study the DNA, so there you can find the DNA when we mention, so it is made up of many different structures. You want me to repeat, Piyush? So do tell me if you want me to repeat this. I'll repeat it again. No for understood or any doubt. I did not get that. So that's why. Okay, so now DNA when we mention, so DNA is basically having three main structures. It is made up of nucleotide. Okay, so nucleotide is there. All right. Any doubt? Nope. Okay, so that's the one. All right. So nucleotide. Okay, so nucleotide it is made up of. So what exactly is nucleotide? So we will see this one. Okay, so now nucleotide when we mention, 1 second. I'll just mute it. Okay, so nucleotide when we mention, so you know nucleotide is basically a polynucleotide together is going to form a DNA. So we can say poly, that means poly means many nucleotides will join end to end forming this DNA structure. Okay, so DNA strand formation we can see with this. And now in this case what all does a nucleotide contain? Okay, so nucleotide will have a sugar. Okay, so there is a sugar, then there is the phosphate group will be there. Okay, so phosphate group is present, and along with that so you can also find that there is a nitrogenous base. Okay, so you find a nitrogenous base. Everything is clear, I'm not having any doubt. Yes, okay, so nitrogenous base will be there. So all of these three together it is called as a nucleotide. So let's see what exactly is this structure now. I mean what exactly is this particular sugar that we are talking about. So in case of DNA the sugar is deoxyribose sugar. Okay, so this is a deoxyribose sugar. That means ribose when we make a pentose sugar. So what is pentose? That means a 5 carbon compound you will find in that sugar. So pentose we can call, and name of that pentose sugar is basically ribose. Okay, so now deoxyribose we are mentioning. So why do we call it like that? So let's see this one. Okay, so now this is the structure. Okay, so this is the structure of the sugar that we will be studying. Okay, so now as usual, so this is that. Okay, so now this is HOH, so different structures you will find. So here also HOH, but here instead of OH H will be there. I'll explain you this part. So then HOH, and here HCH2OH. So this is the sugar, this is ribose sugar now. Okay, so this is carbon 1 position, this is carbon 2 position, this is carbon 3 position, this is carbon 4, and this is carbon 5. Okay, so that is why we call it as a pentose. Pentose is 5. Okay, so 5 carbons are there, so it is a 5 carbon structure. So pentose sugar example is ribose. Okay, and here we are saying it is a deoxyribose in case of a DNA. So what is the meaning of deoxy? So deoxy, D is removal. Okay, so what is removed? Oxygen is removed. From where? From second carbon position. So this is the second carbon position. So from here oxygen is removed. So now both of the on the both the side, so there is HH only. So that is deoxyribose. Earlier this was a ribose sugar. As we have removed the oxygen, so now we call it as a deoxyribose sugar. Okay, so this one is a pentose sugar that we find in case of a DNA. Is this clear, Piyush, about the sugar? In case of RNA, only ribose sugar is there. Okay, in case of RNA, so this was ribose sugar is there. But in case of DNA, deoxyribose sugar. So the previously one that I had drawn with O here, so that is a ribose sugar. So now this one is a ribose sugar. Now if we remove this oxygen from the second position, so then in that case it is a deoxyribose sugar. Okay, so let's go to the next point. So that is about how exactly formation of, so this is about sugar is done. Okay, so next one is nitrogenous bases. So let's see the nitrogenous bases that are there. 1 second. Okay, so let's have a look at the nitrogenous bases. So what are the two types of nitrogenous bases you find in DNA? Any idea? Any of you can also mention. Yes, what is that nitrogenous base in case of DNA? Any idea what are those nitrogenous bases? Okay, I'll tell you this. So now you know we will find two types of nitrogenous bases. So one is purine, all right, and then you also find a pyrimidine. Okay, so there is purine and then you find a pyrimidine. So these are the two types of nitrogenous bases that you find in a DNA. Okay, so now purine, there are two types again. Okay, so one is basically there is adenine, all right, then this one is adenine and this is guanine. Okay, so there is adenine and there is guanine. Pyrimidine when we mention, so here under this, that is mainly in DNA, so we will have a cytosine. Okay, and then a thymine will be there. So there is cytosine and then thymine. So these are the two types that you will find. So purine is there, pyrimidine is there. Okay, so these are the two types of nitrogenous bases that you will find in case of a DNA. Okay, so now under purine, now this one, as you can remember this way, I used to remember it this way, the purine name is shorter. Okay, so with respect to this one, as the name is shorter but the structure is longer here. Okay, so you know the nitrogenous base structure, so it is a double ring structure in case of the adenine or a guanine. So both of these are double ring structures. Okay, that is why it is a purine. Okay, short name but a long structure. Now pyrimidine when we mention, so it is just a single ring structure. Okay, so you can see longer name is there, smaller structure is here, so you can remember anyway, that is possible for you all. Okay, so cytosine and thymine are single ring structures. Okay, so now you know we will see how exactly this purine and pyrimidine are going to make a bond with each other to form a DNA. All right, so in this case, let's see this. Okay, so now this is one DNA strand. So let's say this is DNA strand. So there is another DNA strand over here. Okay, so what exactly is DNA made up of? DNA is made up of a nucleotide. Okay, so it is a polynucleotide chain. It is not just one nucleotide, polynucleotide chain. Now this one is a DNA which is, you know, anti-parallel. So this is 5 dash, this is 3 dash, this is 3 dash or 3 prime, 5 prime also you can mention, and this is 5 prime. So in that case DNA strands are anti-parallel. So what is the meaning of anti-parallel? So one of the strand is running in this direction. Okay, the another strand is running in this direction, so they are running in a opposite direction. So let's say like this. Okay, so they are running in this direction, so they are not running in the same direction, so that means it is anti-parallel. Okay, so we will see what exactly is complementary. So we say that always DNA is complementary. Okay, and we say that the DNA is anti-parallel. So what is the meaning of anti-parallel or a complementary? Anti-parallel, you know that, so one of the strand direction is this way, the another strand direction is this way. Okay, so that we call anti-parallel. Now here you can find a nitrogenous bases that are attached. Okay, so you will find different nitrogenous bases because this is a DNA strand now, so there you can find a nitrogenous basis. So first I'll tell you, okay, what exactly pairs. Okay, so now adenine is one of the nitrogenous base that pairs with the thymine. Okay, so A and T pairing we can see with the weak hydrogen bond. So this one is weak bond and it is a hydrogen bond. Okay, so there is a hydrogen bond will be there, so adenine always makes a pair with the thymine. Okay, so let's see here the adenine is there, so what would be here? So it is with the two hydrogen bond, you can see that there is a thymine that is present. Okay, so now in this case, this we call as a complementary. So what is the meaning of DNA being complementary? So complementary is the base pairing. So now this we call as a base pairing. So now this is a one second. So now this is a nitrogenous base, this is another nitrogenous base. So both of the nitrogenous bases are pairing with each other with 2 hydrogen bond or 3 hydrogen bond. So there is 2 more. Here I was explaining you about a purine and a pyrimidine. So in that case you can find G and C also. Okay, so G always makes a pair with C. Okay, so that is by 3 hydrogen bonds. All right, so in this case, so there is G over here. Okay, so let's say G is here. So opposite end what has to be there? So it always makes a pair with C. So there cannot be T, there cannot be A, or there cannot be G here. So because G is always going to make a pair with C with the 3 hydrogen bonds. Okay, so now G is complementary to C, A is complementary to T, okay, or T is complementary to A, C is complementary to G, okay. So that we call as a complementary. So what exactly is complementary? So when the base pairing is occurring with the respective nitrogenous bases. So A is making a pair with the T, G is making a pair with the T. So between G and C it is 3 hydrogen bonds. Between A and T it is 2 hydrogen bonds. So let's see this one. So I'll just show you this. One second. We will see quiz at the end. So I'll just explain you that. So I'll just explain it here itself. Alright, so now you can see there is one strand. Okay, so now this is a DNA strand. Okay, having a sequence. So here nitrogenous bases are there. So those nitrogenous bases are A, T, G, C, T, T, A. Okay, so let's say this is the complementary base pairing that we can find. Okay, what is the function of DNA? So function of DNA is basically it acts as the genetic material. Okay, so it is a hereditary material that is being passed from one generation to the other generation. Now you know we all have this similarity. So let's say Gurneet is here. So Gurneet looks like her father, or Gurneet is also looking like her mother. So what exactly is the reason? So it is the DNA. Okay, so from your father as well as from your mother, so you have got a DNA. So you have some features, like certain features. So like you might have heard people saying, like nose looks like her mom, eyes looks like her father, hair is like her grandmother. So all these things when we mention, so it is mainly because of a DNA. So DNA from the father, DNA from the mother, so both are basically dropped into a child. Okay, and that child is going to receive some of the external feature from mother as well as a father. Okay, so that is the genetic material or a hereditary material we can say. So that is the DNA. Okay, so now in this case this is the DNA sequence. So we will draw one more here. Okay, now this is a DNA strand. Is this clear, Gurneet? So this is 5 dash direction, and this is 3 prime direction. So 5 prime direction is there, 3 prime direction is there. So now you need to know the complementary base pairing in the other strand. Okay, so what is that complementary base pairing? Anybody knows what is the complementary base pairing for this one? So I'll just write, so you can also write and check out whether the one that you have written is correct or not. Okay, so now this is anyway anti-parallel. That means one is running in 5 dash to 3 dash, and the other one is running in 3 dash to 5 dash. So let's see this one. So this is the complementary base pairing that we need to mention over here. So what exactly is that complementary base pairing? So in this case, so let's see, A is it. Okay, so A will make a pair with T. Okay, so this will make a pair with the T. So that is, this A is complementary to T forming two of the hydrogen bonds. So two of the hydrogen bonds are holding the DNA strands together. Okay, so now this is one DNA strand, this is another DNA strand. So both the DNA strands are held together, that is by a complementary base pairing. Now here T is there, so what would be here? Anybody? So now again as usual, since A and T is them, okay, so here also there is two hydrogen bond. So what would be here? Any of you have any idea? No? Okay, I'll just say. So since T is there, so T is going to make a pair with A. Okay, so A with T, T with A. Okay, so same way now G and C is mentioned. So for G and C, what would be the condition? So this is going to make three hydrogen bonds, and here there will be C. Okay, so this one is the cytosine. So now C is here, so that means again three hydrogen bonds you can see. There is a G over here, so that is complementary base pairing. So what they can usually ask you is, so they'll give a complementary base pairing for one strand, and you need to understand, or you need to write the another strand complementary base pairing. So if ATGC is given, so for A it is T, for T it is A, for G it is C, and for this C it is G. Okay, so this is now a complementary base pairing, only the hydrogen bond variation that we can find. Okay, so now this is the structure that we find in case of a DNA. So let's write this fast. So now T is here, so this is A, T is here, A, again A is here, so this is T, so again T is there, so A, T. Okay, so this is a complementary base pairing, and all of them I have written AT only, so we can just draw two hydrogen bonds for this one. Okay, so two hydrogen bonds, always two hydrogen bonds between A and T, three hydrogen bonds between G and C. So this is G and C, and this is A and T. So this is two hydrogen bond, and there is three hydrogen bond. Okay, so is this clear? Any doubt? Alright, so let's get to the next one. So DNA packaging in case of eukaryotes. Okay, so now one nucleosome can have so this can be about 200 base pairs of DNA can be them. Okay, so nucleosome repeat in a thread like structure called as a chromatin, so that is present in the nucleus. Now further supercoiling of chromatin you can see, so which is forming a loop structures. Okay, so that is of chromatin fiber, so that will form a chromatid. So what is the meaning of this one? So now earlier there you could see there was a nucleosome, so this is a nucleosome now, so here you can find a nucleosome as such. Okay, so now this is a nucleosome which will go on coiling. Okay, so there you can find a coiling there. Okay, so along with this protein, then you find that there is a supercoiling. Okay, so you can also find that there is a supercoiling, so this goes on supercoiling. Okay, so you will see that along with the nucleosome, so this goes on coiling, then supercoiling, again coiling, supercoiling. So you will see that, and as that occurs, so this is going to form a chromatid. Okay, so there is a formation of chromatid. So again you will find a constriction there. So on the other side, so same way there is coiling, then supercoiling, then coiling, again supercoiling you can find. So this is how the DNA is arranged. So now the another chromatid is formed. So now these two are actually chromatids individually. Okay, so individually they are chromatid. So on the whole this is a chromosome. Okay, so this one is a chromosome. So now this center part, what you see, so this is a centromere. Okay, so this is a centromere. So this is also chromatid. So let me just write it. So this is also a chromatid itself. Alright, So now further supercoiling is going to give us a chromatid. So now this structure condenses at the metaphase of the division to form a chromosome. Alright. So what is the total length of a DNA that a human body may have? Any idea? What is that total length of a DNA that a human body may have? So this is not just for one cell. So one cell anyway I mentioned, so that is about 2.2 meters DNA will be there. Okay, so that is for each cell, like we can say per cell. All right, but here it is mentioned total length of a DNA that a human body may have. So it is approximately 110 billion miles. All right. So what is the meaning of that? So if one DNA strand is about 2.2 meter, the DNA from the 100 trillion cells in the body, so they are put end to end. So this would take us to the sun and the back about 600 times. So you can see here, okay. So we are going to the sun, okay, that is with the DNA ladder. So you can see this is a DNA ladder. So what is that DNA ladder? Because ladder we see as there is base pairing, we can see, okay. So that base pairing is forming a ladder like structure, okay. So that is the DNA itself, okay. So now this DNA, when we mention, so question is that what is the total length of a DNA that a human body may have? So in that case, so approximately about 110 billion miles. So one DNA strand, if we are joining, okay, so like from each and every cell, so if we join one DNA strand that is about 2.2 meter, so DNA from the 100 trillion cells, when we put them end to end, so this is going to take us to the sun and back about 600 times, not just one or two times, 600 times. So that is how big a DNA is there, because it is not just one or two cells present in our body. So there are hundreds and trillions of cells that you can find. So based on that, so this is the just the calculation which is mentioned. Okay, so now where is DNA present in the eukaryotic cell? Anybody? Where do you find a eukaryotic, DNA in eukaryotic cell? No idea? So now the DNA is present in the eukaryotic cell. So that is inside the nucleus. Alright, so one second. Okay, so this one you can find inside the nucleus. All right, so next, which of the following comes under the level of organization of a DNA? So it is nucleosome, chromatin fiber, solenoid, all of these. So in the beads on a string model, the bead is made up of 8 histone proteins and a DNA will be there. Okay, so now this is a single human chromosome. Okay, so that may have about million nucleosomes might be there. So now with respect to that, you can see a supercoiling. Okay, so there is a coiling, supercoiling. So this part is supercoiling and this is just a coiling. All right, so this is the one that you see. So now DNA packaging in eukaryotes, when we mention, so I explained you this is the octamer, one at the back, one at the top. Okay, so that is based on that octamer formation will be there. So surrounding that octamer, you can see this structure, that is DNA. Okay, alright, so the proteins in eukaryotes acquire negative charge from amino acid residues with the charge side chain. So that means amino acids which are there in this histone protein, so those are positively charged. Okay, so based on that charge itself, this particular histone protein is having a positive, okay, and DNA is having a negative charge because of the phosphoric acid. Okay, so phosphoric acid is making it negative, or a phosphate group, and histone having a amino acid, so that is making it positive. Okay, so now lysine and arginine are the two types of amino acid which will form a later on a histone octamer, and in general we call it as a nucleosome model, as the negatively charged DNA is coiled around that. So name the scientist who proposed the nucleosome model. Who is that scientist? So it is Roger D. Kornberg. Okay. So the Nobel Prize winner American scientist showed that the nucleosome serves as the general gene repressor of basically with respect to transcription, that is DNA to RNA synthesis. Okay, and prevents the initiation of the transcription. So Don and Ada Olins were the first to discover the structure of a nucleosome. Okay, all right, so before I end this session, okay, so let me just say about certain features of Unacademy. So there is a gladiators batch which started on 6th of April. So here you can watch the recordings that are available already because some of the classes are done. So now from champions batch, this is for those children who are in term 2. Okay, so one second. So this is for those children who are writing term 2 exam. So this also has started. Anyway, you have access to watch the recordings again. Okay, so there is a recordings you can watch. And Hercules batch, it's just started yesterday. So mode of delivery is English here. So if you are planning to take the subscription, you can go for the subscription today. And basically you can go for 12 months of subscription, because since this is just the beginning of class 10, so you can go for 12 months and complete your class 10 with Unacademy, where the maths and science subjects will be covered. So for 12 months this will cost you around 15k. So with the code, that is Vani YT, so this is my code Vani YT. So with this code you will be getting the 10% discount on the subscription. So here the mode of delivery is English, here the mode of delivery is English. Okay, so you can go for any of these batches based on your comfort in studying, like in particular language. Okay, so you can go for any of these batches, and don't forget to mention a code, because only with that you will be getting a discount. So introducing community, never miss on any update from your favorite educator. Get session notes and practice material. Share your feedback directly with the educator via poll. Okay, so check out the link to the educator community below. Okay, so Bugs Bounty, so this is opportunity for all learners to report any inappropriate content during the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Alright, so how you can watch my special classes? All you need to do is download the Unacademy Learner app, open it, then you can enter your mobile number and OTP, then sign up using your email ID. So once you do that, you can select class 6 to 12, then select your category ICSE, select your preferred language like English, Hindi or English. Go to the description of this video, click on the special class link, then click on the notify me to get notified about the class 10 minutes before the class begins. You can also use my code VANIYT to unlock the free products that are there on Unacademy. Also, you can use this code to unlock, to get 10% discount on the subscription. Alright, so see you all in the next session. Thank you everyone. Any doubts you have, you can ask. Any of you have any doubts? Alright, so yeah, so click on bell icon to get notified about the classes. Subscribe to our channel, that is Unacademy ICSE and ISC both. Like and share the video and download the Unacademy Learner app. So this PDF, anyway, I can send it on my Telegram. So that is Vani Kari ICSE class 9 and 10 biology. So you can just join this Telegram. So I'll send all the PDF that are there with me for this class. Okay, so you can join this Telegram. All right, so see you all in the next session.